cities and the calculating portion. I feel like that feels a little late. So like, I feel like I should have probably tried a little harder to get more earlier, but we'll see how it shakes out.
Sometimes they're not very hard to do, especially if you're concentrating on something.
Right. I mean, that's always, that was always one of my ideas of like always just have 3 cities early game, and then if you're going to go to the elimination path, just take the fucking cities as you go. You have 3 cities. Now, in Cigarette, they don't have like the opportunities for like the 5 turns or whatever, like the city just overplaying and doesn't make anything. Is that a thing that doesn't still? Okay, yeah.
Although it makes more sense in other cities.
All right. As we begin.
Yummy.
I'll be on amplified back.
Be like, hey, I noticed half you assholes weren't here yesterday. $5 for them.
Make an anonymous email,
For sure. Put a paywall, give them the first half hour, and be like,
Testing, I think I'm amplified. Okay, so item number 1. I finally have your exams graded, and I'll go into details about why. But so over the weekend, I'll work it all up and get it posted for you. Apologize for the delay. But this class is getting ever larger semester by semester, or year by year, and it does take time to grade the essay questions. I'm blaming my teaching assistant. So we move on now to talking about more about B cells. Now we're going to come back and talk more about T cells, and we're going to talk about the T cell receptor in more detail, and also the MHC presenting protein, which is expressed on the surface of an antigen presenting cells or virus infected cells. So you remember we said that the T cell receptor is evolutionarily related to one of the antibody domains, or the FAB fragments, or FAB domains here, one of the arms of an immunoglobulin molecule. And we have an alpha chain and a beta chain. The alpha chain is related to the light chain of the arm. The beta chain related to the heavy chain component. So let's take a look at more detail of what a T cell receptor actually looks like at the level of crystallographic structure. As we know already from our discussion of immunoglobulin molecules, we have the so-called immunoglobulin fold here in the constant domain of the TCR alpha chain, and then also the variable domain of the TCR molecule. And same thing for the beta chain, okay? Here, what we when we generated an antibody, we talked about how we have a preformed gene for the maybe light chain constant domains and the heavy chain constant domains. And then we have a variable sequence which is composed of a set of pre-made structures like this, and then certain hypervariable segments on the gene that are going to be assembled to make the proper gene. We talked about that in the case of antibodies. I'm going to go back and remind you about all this. And when we talk about assembling the gene for a T cell receptor, we'll play to this this afternoon. But you remember, there are hypervariable regions of both the variable and heavy chain, of both the light and heavy chain components of both the antibody and the T cell receptor. And here are these hypervariable amino acid sequences. They take the form of these loops here at the crest of the variable immunoglobulin structure here. And it's these variable loops here that are going to then interact with a combination of the foreign peptide and the MHC presenting molecule. We'll be discussing again in more detail. The crystallographic structure it did not include the transmembrane segments here and the very short cytoplasmic tails. So this would be a T cell. This would be its surface membrane. These are the transmembrane hydrophobic 20 amino acid transmembrane segments here, and then very short cytoplasmic tails, even shorter than I've actually written. And so, continue on here. So, as I just said a second ago, just as is the case with B cells, the diversity of T cell receptor proteins, okay, comes about during T cell development from a stem cell precursor that's going to leave the bone and go to the thymus, and then those cells are going to be growing and differentiating, and finally they will have produced a clonal descendants of T cells expressing a proper T cell receptor on their surface. And again, by analogy to the antibodies, we have here the alpha chain, which is evolutionarily related to the light chain of an antibody molecule, has just V boxes and J boxes, okay, and then a constant part right here, which is going to be this part of the protein down here. So the DNA sequence that will encode the constant domain of the alpha chain, that's pre-made and all cells have it, but they don't express it, except in T cells. We will express it after we have picked a particular V box and a particular J box and brought them close together at the level of DNA, and now we can make that RNA here, which will code for the alpha chain protein. And similarly with the beta chain, which is analogous to the heavy chain of antibodies. For heavy chain, you remember we have D and J and V boxes, and we pick randomly from the menu here and create a finished gene at the level of DNA for a particular beta chain of a T cell receptor, and then we'll make the mRNA and protein out of that to make this beta chain. And again, by the way, just as was the case for T cells, we insert these random P and N nucleotides every time we cut, from a segment here to a segment here, loop out the intervening DNA, cut, and then before we ligate this B and J, let's say together, we insert random nucleotides, P and N nucleotides to generate, mountain of stars of the universe type of diversity possibilities here. Now, the T cell receptor, there's a bit of a semantic ambiguity here. Whenever I ask you exam questions about this, I'll try to make clear what I mean here. We speak of the T cell receptor, but actually in T cells, this is always found together with partner proteins. Okay, a set of proteins, 4 in in number, 4 different kinds of proteins, but two of the epsilons, a delta, a gamma, and then the zeta chains here, a pair of zeta chains here. So we have yellow, red, and green accessory proteins here. Now, this together is sometimes simply called the T cell receptor, meaning which, depending on the context in which we're saying this, when we say T cell receptor, we will often mean this whole complex here. On other occasions, depending on the context, we mean just a pair of blue guys here, the alpha and beta T cell receptor per se. So you'll never get confused about that. The cytosolic tails of the T cell receptor are really quite short, a couple of few amino acids kind of thing. And so the blue proteins, the cytosolic tails, they're too short to recruit signal transduction molecules. And the other chains here, red, yellow, and green, they then actually are what recruit the signal transduction molecules, such that when this T cell receptor in blue recognizes the antigenic peptide presented by an MHC molecule, we will then activate, things will happen inside. I will not spare you the details of the things that happen inside, but that's another lecture down the road. Things will get phosphorylated here, and they will recruit signal transduction molecules, initiate signal transduction pathways that in many ways closely resemble the signal transduction pathways that we initiate when growth factors bind to a growth factor receptor, for example, please-derived growth factor binding to the PEGF receptor, and paternal growth factor binding to the EGF receptor. T cell receptors binding to their peptide. We initiate these signal transduction pathways that lead this cell to proliferate and also to differentiate. Now, this is not going to be, this is one of the signals, but remember we're going to have other signals that are going to be required for this T cell to give the full response of proliferation and differentiation. Okay. Now, the red and yellow guys are known as the CD3 antigen, or sometimes the CD3 complex. Okay. We didn't know what this thing was. It was just a cluster of differentiation number 3. We identified all these things as unknown. We didn't find out what they were. Now know that the CD3 antigen is this set of proteins here. Now, the zeta chain was not detected in the methods that we use to detect the delta and epsilon molecules here. But I think I'm correct in saying that the red, yellow, and green guys are basically the CD3 complex. Okay. Because there's some ambiguity in my own mind about whether zeta is part of the CD3 antigen, I won't ask you any question about that in the test, okay? But if I speak of the CD3 complex in a test question, I'm referring to all this stuff here, basically. Okay? The signal transducing complex, basically. Okay. Now, things have been nice and simple so far. We say the T cell receptor has 2 chains, alpha and beta. Well, I'm sorry to tell you that there's another set of T cell receptors known as gamma delta T cell receptors. So 95% or some number like that of T cell receptors are alpha beta receptors, the classic ones that we first knew about and I'm teaching you about. At a little bit later date, we realized that there's another subfamily of T cell receptors, and it's not alpha and beta chains, but gamma and delta chains, and they have their own genes, which are assembled kind of like, I think, the alpha and beta genes. Maybe not, actually. The gamma delta genes, I beg your pardon, yes, in fact, they are assembled that way. So, in addition to alpha beta, we have gamma delta. Just as in the alphabet, you say A, B, C, D, A, B, alpha, beta. D, E, F, G kind of are like, we say gamma delta in the Greek alphabet like we say AB in the Latin alphabet. So gamma delta, gamma corresponds to the alpha chain of a T cell receptor, delta corresponds to the beta chain. So that the gamma component here, again, like alpha, resembles the light chain of an antibody, and the delta subunit here, which would correspond to the beta chain of an alpha beta receptor, and like the beta chain, it corresponds to the heavy chain of antibodies. Okay, so I've written that down here as well. Do you have some of the stuff in your notes? I have somewhere here. The gamma corresponds to alpha and alpha beta TCR, which corresponds to the immunoglobulin light chain. And the delta chain corresponds to the beta chain of an alpha beta T cell receptor, which corresponds to the heavy chain of an immunoglobulin heavy chain. Okay. Now then, let's just read this together here. So, there is a second type of T cell receptor, similar in overall structure to the alpha beta, but is formed of two different proteins, gamma and delta. T cells with gamma delta receptors arise early in embryonic development, and they play a role early in life, early after birth, but are eventually largely replaced by cells expressing the more versatile repertoire of alpha beta receptors. Gamma delta receptors are thought to represent an older, more primitive, and somewhat less specific component of the adaptive immune system, with aspects of innate immunity kind of in it, basically. Okay? The gamma and delta loci, like the alpha and beta, consist of sets of V, D, J, and C segments and so forth. And they're going to be assembled in ways that we'll talk about just briefly. Okay, so now you're not going to have to remember chromosome 14 and all this stuff here, but I'll just tell you that for the gene that encodes the alpha chain of alpha beta TCR, in between the set of 75 or so V boxes, one of which we'll choose to make a particular TCR gene, and the constant part of that gene, we have a whole set of segments here that are used to make delta chains. Okay, so this is the kind of a beta chain like gene that we assembled in the middle of an alpha chain gene on chromosome 14. And then on chromosome number 7, we have the various components we will use to make the gamma, a gene encoding the gamma subunit, gamma delta T cell receptor. We'll talk in more detail here. We first make it a VJ pair. If that gene doesn't produce a functional T cell receptor, we have a second go. We'll scrap this and then grab one of these J's, join it to another C component, and then compare the proportion to V box. Grab a V, pick a J, and then this V we'll pick with one of these J's, and then we'll have a second shot at the constant domain. We're going to be going into more detail. I think I've already mentioned in the case of B cells in the bone marrow, as they're going from a stem cell precursor to a complete B cell making an immunoglobulin M on its surface. It is a brutal Darwinian process. Many are called, few are chosen, so to speak. You assemble a gene, and often this just doesn't work, or it's against self or whatever, and so lots of stem cells never end up growing up to be a functional B cell that goes out to the bone marrow, and the same thing is true with T cells. Now, again, just to remind you, we said that the alpha beta T cell receptor doesn't bind the surface of a whole protein. It can only bind to peptides derived from a foreign protein that are being presented on an MHC molecule. So here's an MHC molecule. Here's an alpha beta T cell receptor. Okay, and the tips of this receptor have these hypervariable loops, which are then going to both alpha and beta chains contribute to the tight binding of the T cell receptor to the foreign peptide MHC complex. Okay. Well, let's take a look here. Just a second. This story. No one reminded me to sit down on my stool. I need to be reminded because I'll go on for too long before I'll pay the price by the end of our 3-hour lecture. I know you're all very interested in my health problems. Okay. So, here is an MHC1 molecule presenting a peptide. Now, is this peptide going to be derived from extracellular bacterial foreign proteins? Or is it going to be derived from intracellular viral proteins if it's being presented by class 1? Intracellular, right? Now here's the class 2 MHC presenting, of course, now you know it must be this one is presenting proteins, peptides derived from extracellular proteins that have been brought in, cut to pieces, and more of the peptides loaded onto this class 2 molecule. Only a CD4 protein, only the T cell receptor on a CD4 cell is able to bind productively a peptide being presented by class 2 molecules. And that's mostly because the CD4 protein here plays a role in this whole TCR peptide MHC complex here. The T cell receptor, these transmembrane segments of that have the properties such that they spend their time, let's see, be honest now, how many of you have never heard of a lipid raft subdomain in the plasma membrane? Lipid rafts in the plasma membrane and some of the intracellular membranes. They are little islands of a special sort of set of lipids, rich in cholesterol and also rich in sphingolipids, and they're a little bit thicker than the phospholipid bilayer of the bulk glycerol, phosphoglycerol type lipids. Okay. How many of you do I need to say a little bit more about this? How many of you are not so very familiar with lipid rafts? Okay, thank you for your honesty. So I will take a moment. Well, I pretty much said what I wanted to say. I don't know, it was probably 20 years ago or more, we discovered that the bulk membrane is the sort of phospholipids, you know, phosphatidylethanolamine, phosphatidylserine, and so forth, that make up the bulk phospholipid bilayer. But then there are other membrane lipids, as I said, generally sphingolipids, which have longer hydrocarbon chains, and cholesterol molecules, which form clusters. They're kind of evanescent cultures. They assemble, maybe get evaporate and then they reassemble again, and they can come together to make larger islands of these patches on the surface of the cell. Certain proteins preferentially partition into these lipid raft structures, and if they happen to diffuse out, they're not so happy. They expose hydrophobic side chains into the aqueous environment, and they pretty soon will find their way back to a lipid raft and be happily there for a long time. And then certain proteins preferentially insert into these thicker phospholipid rafts. The T cell receptor is one of these proteins that prefers to partition into a lipid raft, as also the CD3 complexes, which are not shown here. Okay? Now, the CD8 and the CD4 protein also prefer to live in lipid rafts. So whenever you have a T cell receptor, these, in the absence of when the T cell receptor is not interacting with an MHC molecule, but just all by itself in the membrane waiting for something to happen, there will be either a CD4 protein or a CD8 protein, depending on whether it's a CD4 or CD8 T cell, not very far away, because these lipid rafts are about maybe I'm a T cell receptor and you're a CD4 protein. We live in the room here. We're never too far apart from each other. Maybe we live in the first few rows here. So that when we form this complex between the T cell receptor and its ligand, it's only a matter of a few milliseconds before, just by chance, this will come cruising along, bump into this, and the tip of the CD4 protein tightly binds to the base of the T cell receptor. I'm sorry, to the base of the MHC molecules here. Now, T cell receptors have constant domains here. MHC molecules don't have constant domains. Okay, it's just beta 1 and beta 2 domains, and they don't resemble the immunoglobulin fold or anything like that. But the CD4 protein here sticks to the side of the beta domain number 2, or the beta 2 domain like this, and really stabilizes this whole complex. Furthermore, when the CD4 protein, its tail, as I'll be discussing in a subsequent lecture in more detail, there's a Stark family tyrosine kinase that associates with the cytosolic tail of the CD4 protein and also the CD8 protein. So that as this thing is diffusing around, as soon as it bumps in and sticks here, it brings this Stark family tyrosine kinase right up next to, sort of kissing right up next to a protein with a phosphorylatable tyrosine residue on it, and that is the beginning of the signal transduction events that are going to be happening. So CD4 T cells ignore MHC1 molecules because they don't have any CD8 proteins, and the CD8 T cell, which does have CD8 proteins in a way that was analogous to CD4, CD8 proteins bind to the base of this MHC molecule here. So CD8 proteins stabilize a T cell receptor MHC1 peptide complex, and again bring Stark family tyrosine kinases into play, initiating the signal transduction event we're going to follow. So CD4 T cells recognize only class 2 molecules because they have the needed partner protein, CD4, which can only bind the base of MHC2 peptide presenting complex. Yes?
So can you just quickly go over again the intracellular, or not intracellular, intramembranous part of the CD4 interacting with the beta chain of the TCR?
Well, I said that, there's a lipid raft, which is kind of like maybe like this or something, and the CD4, in the absence of the T cell receptor binding to its foreign peptide, the CD4 protein is just wandering around the surface of this little island in the South Pacific,
You mentioned something about signal transduction.
Yes. I said that the CD4 protein brings with it at its base a SARC family tyrosine kinase. Again, I'm getting way ahead of myself. We're going to have a whole lecture on the signal transduction stuff. And many of you have no idea what I mean by a SARC family tyrosine kinase, I imagine as well, but no, just out of curiosity. Who of you kind of know what I mean by a SARC family tyrosine kinase? Okay, good. That's the Australian say, good on you. We'll be talking all about that. Okay. These are more or less the structures of the CD4 and CD8 proteins here. I'm not going to have to do that. The CD8 protein you might think would be kind of floppy here. I don't believe these are alpha helices here, but one way or another, they're probably not just vertically erect. They probably have some kind of folded structure on them. But I think they're emphasizing domains here. They can, well, whatever. I'm just showing you what's known about the structure of CD4 and CD8. I won't ask you any questions on the test about what is the structure of CD4 and CD8. They don't have any of these even globulin fold bread and butter sandwiches that I talked about, frozen for structures. The HIV virus has the viral spike proteins on the surface of that virus. They specifically bind the CD4 protein, okay? And then they roll over and bind another ligand expressed by CD4 T cells, and they then infect a CD4 T cell and kill CD4 T cells. And after years, you can replace CD4 T cells that have been killed by the virus for a period of time, but after 5 to maybe 10 years, you've exhausted your ability to replace the CD4 T cells that the virus is killing, that's the Achilles heel of the whole immune system against this kind of thing. So the immune system collapses, and then you die of the next little pathogenic virus or bacteria that comes along. Okay, so again here, CD8 T cells are going to be ultimately killer T cells. We say that these CD8, we say that we say that the T cell receptor on these CD8 T cells, the early immunologists who were beginning to sort of sort through and figure out what was going on here, they would use a phrase that CD8 T cells, or the CD8 T cell receptor, is MHC1 restricted, by which they mean it can only respond to peptides presented by an MHC1 molecule. Okay, and if they would see that, they would either then become a killer T cell, or if they were already a killer T cell, they would respond by destroying the cell that was presenting this peptide. And similarly, we would say that the T cell receptor on CD4 T cells is MHC2 restricted. Okay, this T cell receptor can only recognize foreign peptides presented by a class 2 molecule. Okay. In this cell, we may have peptides being presented by class 1 molecules, but this CD4 T cell can't see those peptides. It can only see the foreign peptides presented by class 2 MHC molecules. Maybe I'll pause for a second to let that sink in. We'll be returning to that idea. Okay. And of course, the CD4 T cells, if they encounter a foreign peptide presented by a class 2 molecule, they will then proliferate and differentiate into either T helper 1 cells or T helper 2 cells. Okay. So let's take a look now at the structure of these MHC molecules. Here is the structure well, here's the cartoon structure of the class 1 molecule, which we've been talking about. okay? It'll be holding, we're looking kind of in cross section at the foreign peptide. My pencil would be the foreign peptide, and we're looking at a cut through my pencil here. That's what that little ball is. So this is a long groove here, a little valley in which this long peptide pencil is sitting on the floor of that valley, which is created by this structure right here. And here's the T cell receptor. The loops here on both the alpha and beta chains are recognizing epitopes on the peptide as well as epitopes on the MHC molecule here. So the interaction of the T cell receptor, it's not just T cell receptor foreign peptide, it's T cell receptor peptide MHC complex. And that was an important complex. That was an idea that took some decades for immunologists to begin to understand. That's why MHC1, why CD8 T cells, that's why they are class 1 MHC restricted, okay? They can only see the peptide, and they make sure it's a class 1 molecule here, in part because you can't get tight binding, but also, as I was emphasizing, because these cells express CD8, the CD8 can get recruited and bind this domain right here, which is kind of immunoglobulin related. I was saying that the CD8 protein doesn't have immunoglobulin bread and butter sandwich but this part of the MHC molecule does have little bread and butter beta sheet structures, a classic immunoglobulin, well, kind of a disorganized bread and butter sandwich structure. Okay, and now for the class 1 molecules, a single polypeptide chain makes up part of this base structure here, and then also this platform that the peptide is on. Class 2, as I'll show you in a moment, it's the alpha and beta chains here of a class 2 molecule, where alpha contributes half of the structure, beta contributes the other half of the structure. For class 1, we need something to support the platform, and there's a sort of a general purpose protein here called beta-2 microglobulin, which will come in here, slot right in, and then interact with both this and this part right here to kind of stabilize and keep sort of not folding downwards, keeping it oriented perpendicular to the T cell receptor axis. And this, of course, would be typically a virus-infected cell because it's intracellular viral proteins, which are peptides are created from those foreign proteins that loaded onto the class 1 molecules. And here's the class 2 structure. Again, as I said, in this case, the platform on which the peptide is sitting here is composed half of alpha subunit protein and half of beta subunit proteins here. We don't need no beta-2 protein in here. Each of these chains provides its own sort of base supporting structure for the platform that the peptide is going to be. Okay. And it's not really so clear here, but in both the class 1 and class 2, what we have here is kind of a one alpha helix, if this were a sheet of paper, one alpha helix here, one alpha helix here, and then this is a flat beta sheet platform floor here. So the peptide lies in between the two alpha helices and sitting on the floor of the platform. Okay, I'll show you more pictures of that if that's not quite clear. Okay. So here is a clearer picture. Here is the alpha helix. Here's a second alpha helix. This is a beta sheet here. This is the floor. This is like a little valley here. Here's the mountains. Here's the mountains. Here's the floor of the valley. Maybe there's a little stream running down the floor of the valley. But it's a flat surface, and the peptide sits right on that flat surface, and it interacts with the side chains of the beta sheet floor of the valley, as well as with side chains of the alpha helical walls or mountains of the valley. In the case of class 1 molecules, the valley is closed at either end. So the peptide, it can't be longer. If the peptide gets to be this, if the peptide were to extend like from here to here, it wouldn't be able to fit in because it doesn't fit inside the floor, the walls of the mountains here. But for class 2, the valley is open-ended. So you can have long peptides that sit here and then drape over or leave it free at either end of the valley. So we have peptides of fixed length that can fit in the peptide binding groove of class 1, but we have peptides of a wide variety of lengths that can be presented by a class 2 MHC molecule. And I have a picture of that too. So here is the MHC2 Happy Valley here, the mountains and the platform. Here's one of the peptides here that fits in. Now, if the peptide were fully extended and lying flat on the valley, it could only be 8 amino acids long. Okay? But it can be up to 10 amino acids long, actually, because we could have it lying flat, 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 and then buckling up, and then coming down, being flat, maybe buckling up again and being flat. Okay? So it can be a little bit longer than 8, just because it can, one amino acid can kind of buckle upwards. So 8 to 10 you can just remember 10 is an easy number to remember, but actually it's maybe more optimal to have it a little bit shorter than 10. For the class 2 molecules, we can extend outside the little Happy Valley structure, so you can have anywhere up to 25 amino acid peptides. So terminal end to carboxyl terminal ends, one can be long, one can be short, they could both be kind of long, they're just sort of waving free on either end of the Happy Valley structure. So that gives class 2 molecules a better chance of there being a peptide that they can bind and present. For the class 1 molecules, there's a little bit, there's a restriction here. It has to be 8, the peptide has to be 8 to 10 amino acids. Now, I want to take a break now. And when we come back, I want to talk about the concept of anchor residues or anchor amino acids. So we'll start this after we take a stretch here.